This afternoon we're starting with Marie Manceau, in fact. As you can see, she's working on stripes in birds and mice. And other animals? No. Penguins? Yeah. And well, as we had this morning, I hope we have plenty of discussion. Thank you, Marie, for coming from so far.
Well, good afternoon, everyone, and thank you very much, Alain, for this very nice opportunity to speak about the work that we are doing in the lab. So we are interested, I guess, like many people in this room, in biological patterns, which we can define as the visible order in the spatial organization of characters across the surface of an organism. And these patterns, they form through a series of highly orchestrated steps during embryonic development. First, we start with a homogeneous tissue, and this tissue is going to acquire positional information. This will give rise to coordinates, according to which cells will adopt specific fates and differentiate to create a pattern phenotype. So what is stunning is that there is a huge diversity in these pattern phenotypes in nature, such as, for example, in the distribution of appendages across the surface of the skin or in the distribution of limb elements. So really the question that has been fascinating, I should say, both mathematicians and developmental biologists for more than 50 years now is what are these pattern-forming events and how do they underlie this stunning diversity in patterns? And I think one of the patterns that has raised a lot of interest is the periodic patterning of the coat color in repetitive motifs such as stripes or spots. You can see that sometimes even within one species, there is a lot of variation in these periodic patterns, from small spots to big spots to stripes. And this has been intriguing far before the realm of mathematics and biology. But despite this interest, we don't know much about how these periodic patterns formed, and this is due to two reasons. First, we don't really know what is the histology of the skin tissue that is organized in a periodic pattern. We don't know what's going on in the skin of a zebra or of a leopard. And second, we don't know much about what gives rise to positional information. So we would need to know which type of molecules are involved. And of course, morphogens are excellent candidates because we know that many of them diffuse at long range in the developing skin, but they are expressed in gradients that do not necessarily spatially correlate with the adult pattern. So it's difficult to make a causal link in the absence of spatial reference. What is also highly debated is the origin of these positional signals. Is the skin organized through an instructional source, a signaling source, or does it self-organize according to dynamics such as Turing's reaction diffusion? And finally, recently, there has been a lot of emphasis on the role of morphogenesis, not only into integrating positional information, but also actually into generating this positional information. So in the lab, we study all these different aspects, and we use two predictive methods. The first one is fairly classical. It's been used widely. It's a theoretical framework, so we generate computer simulations of stochastic dynamics, such as, again, reaction diffusion or chemotaxis, or a combination of those. And we run simulations to reproduce patterns that we see in the wild. So this is not work from my lab. This has been done many, many times to reproduce the stripe pattern variation in the fish mutant strains, for example, or in different organs in the fish or in mammals. And the second method that we use, and perhaps it's a bit more specific to what we're doing, and I will really focus on that approach today, is we use natural variation as a tool. So instead of just describing one periodic pattern, we compare several. And doing this, we try to find common points and differences that we link to common points and differences in the developmental events, in the hope that this will help us predict which of these events are involved in the patterning. So I will talk about the work of Nicolas Hoppé, who joined as a postdoc about 3 years ago in the lab. And Nicolas has been interested in the striping pattern that we can observe in a group of birds called the galliforms, which you can see here. These are pretty much what we call poultry, so foals and game birds. And what's interesting with these guys is that when they are just born, as chicks, as juveniles, they display a striped pattern along the dorsum. So you can see several of these species here. And if we choose one, the Japanese quail, you can see that there's an alternance of longitudinal stripes, black and yellow stripes, across the surface of the dorsum. So the first thing that Nico did was to take his car and drive around Normandy and Brittany in France and go shake hands with farmers that are raising these animals for the food industry or for hunting purposes. And so he collected many specimens of different species, and you can see here these specimens in a slightly flattened form. So on these flat skin preparations, you have a view of the back of the animal. Here you have the wing, and here the legs. Okay? So the first thing that you can see is that in all these species, the pattern is symmetrical over the body axis, so over the dorsal midline. And then there is some sort of conserved stripe sequence. In all the animals, we observed that the central stripe was black. So sometimes in some species, it is split by a few yellow feathers that give rise to very thin central yellow stripe. But most of the time, really, it's a central black stripe that is flanked by lateral yellow stripes, themselves flanked by dark stripes. So there is a conserved black, yellow, black sequence from the dorsal midline. And Nico has quantified this sequence by measuring the distance from the dorsal midline to the boundary between each stripe at the level of the wing and the level of the leg. So he got this type of results in all the different species. And the first thing that I would like you to see here is that very often in the leg, in which the skin is wider, there are more stripes than in the wing, in the region in which the skin is thinner. And also we have often more stripes in larger animals, such as the blue-eared pheasant or the wild turkey, compared to smaller animals, such as the Japanese or mountain quail. So what this tells us is that with dorsum size, there is an increase in stripe number. So the variation in the pattern is not due to simple scaling of the same motif. And then the second observation that Nicolas made is that when he looked on the centralmost stripe, so the central black stripe or the lateral yellow stripe, he could see that independently of the size of the dorsum, these stripes were always kind of in a comparable position. But on the contrary, their width vary a lot. So comparable position, variation in width. So we wanted to go further into this phenotypic description and compare the striped pattern to the pattern of the feathers. Because I don't know if you know, but in birds, feathers are not distributed throughout the whole body. They are organized in what we call feather tracts, which are regions in which they are implanted, and they are separated by near glabrous regions. So these tracts exist in galliforms. So here again, you can see a dorsal view of flat skin in the Japanese quail, from which we plucked out all the feathers to reveal the implantation of feathers. So here you can see the dorsal tract on which we are focusing on. And within the tract, feathers form these longitudinal rows that we can really number from the dorsal midline. So feather row 1, feather row 2, 3, and so on. And what was really cool is that the number of these rows and their spacing varies between species. And it's typical to each species so that we have really a map, a reference map of the feather tract for each species. And on this map, Nicolas has mapped the color pattern. So if we follow here this line in the Japanese quail, so the wing would be here, or just under the level of the wing. Let's follow this line. We can see this color code on the feathers. Black, black, yellow, 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 black, black, yellow. So that code, he then reported on the whole dorsal tract to obtain this type of maps. And he did that for all the different species. So here I'm summarizing a huge amount of work. But bottom line, what Nico could see is that the boundaries between stripes are always located in the same position within one species. For example, that boundary here in the quail is always located on feather row number 2. But this boundary varies between species, so that boundary is always located on feather row number 7 in the mountain quail. So from these observations, we thought, okay, maybe the periodicity in the striped pattern could be linked, could be due to the process of feather tract formation. And that was a very appealing hypothesis because we knew that this process occurs in a morphogenetic wave. So this was work done by Richard Bayeul, who is a grad student in the lab. And what Richard did was to stain Japanese quail embryos at different stages of development with beta-catenin, which marks the developing feather follicles. And so what he could see is that the tract forms first through these 2 longitudinal lines that will form the first feather rows, and then each feather row is added laterally, one by one, in a sequential manner, until the completion of the whole tract. And then once the tract is completed, then we have a second wave of pigment appearance. So first, in this area here in the middle, you can see that we have first the apparition of the black pigments. And then, I'm sorry, it's not really clear here, but here laterally will later appear the yellow pigments. And then during the late phases of differentiation, the whole surface of the skin switches back to black pigment production, so that in all feather types, whether they are black or yellow, they all do have a black basis. So what Richard has shown here is that there are 2 sequential waves, medial to lateral waves, in feather tract and pigment production. And during this wave, there is a pulse of yellow production localized in a band in the skin. So the question was, is that pulse triggered by a certain stage of tract differentiation? So to answer this question, Nicolas has compared the situation in the Japanese quail with what happened in 4 other species that we chose for 2 reasons. The first one, because they displayed variation in their color pattern. The second one is because they are used a lot for the food industry in France, and so we could get a lot of fertilized eggs and thus embryos. Kind of pragmatic reason. And so in all these 4 species, we saw the same thing as in the quail. The pigments are produced in a medial to lateral wave. And during this wave, we have yellow pulses, localized yellow pulses of production. Okay, so here, again, I'm summarizing a huge amount of work by Nicolas and Richard, who have described in all of these species the dynamics of tract formation, feather row by feather row. So you can see here the result. I've color-coded the scheme with the first form rows in dark blue and the last form rows in dark green. So this color gradient represents the wave of feather row formation. And when we link this, when we compare this to the position of the yellow stripes in all the different species, we don't see really any correlation. So it seems that, at least from this comparative analysis, the yellow pulses are not triggered at a certain stage of tract differentiation. So we thought, okay, maybe it's a certain stage in the feather growth that triggers the yellow pulse. So Nico has compared the developmental stages of feathers in all these species when they first start to produce yellow, or when they switch back to black pigment production. And he saw that this occurs at different stages depending on the species, which gives rise to different length in that yellow band along the feathers. So again, no correlation. It seems that the yellow pulses are not triggered at certain stages of feather growth. So I have to say at this point, we were a bit disappointed because it was really cool to think that the periodicity of the stripe pattern was linked to the periodicity of the tract formation. But we couldn't find any nice correlation, so we switched gears. And instead, we started to think maybe there is a pre-pattern prior to the formation of the tract acting in the skin. So Nico has screened for the expression of many, many candidate factors, and he found one that was interesting. This gene encodes a signaling peptide called agouti, whose function is to have pigment cells switch from the production of black pigment to the production of yellow pigments. So pretty much when agouti signaling is active, you make yellow. And this was a good candidate gene because I had shown during my postdoc in rodents that this gene was expressed in the ventrum, restricted spatially, and in this area it was able to contribute to the patterning of color. So what Nico has shown is that in birds it's not only in the ventrum, but it's also in the dorsum, and it is expressed in these stripes. So you can see here in the Japanese quail that there is one medial stripe of agouti expression, flanked by two intermediate stripes of agouti expression, themselves flanked by two lateral stripes. That's the case of the Japanese quail. In the partridges, we have one thicker medial stripe and two lateral stripes, and in the pheasants, only two very large lateral stripes. And what was really nice is to compare this expression pattern to the position of I'm trying hard to the position of of these yellow feathers, and you can see that this was really corresponding. So agouti may correlate with the yellow striping pattern. So to confirm this spatial correlation, Nicolas has performed double in situ hybridization for both agouti and beta-catenin at different stages of the quail development, because by doing so, he could define which feather rows have expressed agouti in their presumptive domains. And so what he could see is that agouti had been expressed in here, these central feathers, which form in the area covered by the medial line of agouti expression. Feather row number 3 exactly corresponds to the intermediate line of agouti expression, and feather rows 4, 5, and 6 corresponds to the lateral line. And so these feather rows are those that later produce yellow. So really the expression of agouti foreshadows the position of yellow stripes. But so far it was only a correlation, so we really wanted to make the functional link, functional experiments, and so for this we were helped by David Goéchon from the INRA, who provided strains of quails that are mutant for agouti. These strains were existing, we were kind of lucky. And one of these strains is called the recessive black. You can see that it's much darker than the wild type, and it was shown that in these mice there is a decrease in the levels, the overall levels of agouti expression. But we showed that the pattern of agouti expression is conserved. And in these quails, we observed that there was a disappearance of the central stripe, and the lateral yellow stripes were much thinner. And Nico actually was able to show that they appear thinner because the feathers in this area produce yellow for a shorter period of time. So decrease in agouti levels form thinner stripes through shorter yellow pulses. Conversely, in quails in which the expression of agouti is increased, these are called the fawn mutants, we could see that the pattern of expression is conserved, but the stripes are larger, and this was due to an increase in the duration of the yellow pulse. So together with this loss and gain of function experiment told us is that first they confirmed that agouti establishes a pre-pattern of yellow production, and that it acts by controlling the duration of the yellow pulse along the feather. So, and this occurs in a dose-dependent manner. So if this is the surface of the skin, here you have the dose of agouti expression locally. At a certain threshold, the cells that receive agouti signaling will produce yellow. Now, if we increase levels of agouti expression, at that same threshold, we will have a longer pulse of yellow production and thus a wider stripe. And on the contrary, if we decrease that expression at that same threshold, we'll have a thinner stripe or no stripe at all. So we think this is the way this is working in these quails, showing that agouti controls the width of the stripes. And what we are studying now is the expression levels of agouti in all the other species, because it would be really nice that if it was as simple as all these species evolve stripe width differences simply by regulating, by changing the expression levels of agouti or the threshold at which the cells respond to its signaling. So we're studying this currently. So that was for stripe width, which varies a lot between species. Now, what about stripe position? So if you remember, I told you that the position of the centralmost stripe is comparable between species, which led us to think that maybe their pattern is set early on, before the late development of the lateral parts of the skin. And if that's the case, what could be the signaling instructional, if you wish, the signaling source? And here we went back to old experiments that were performed more than 30 years ago now by Nicole Ledouarin and colleagues, where they grafted neural tubes from quail embryos in place of those of the chick embryos. And by doing this, they obtained chimeras in which, in the area where the neural tube was grafted, the chimera displayed the color of the quail. So these results showed that the neural tube is at the origin of the pigment cells. This is where this is the origin of the lineage of the pigment cells, and is responsible for coloration. So it was the obvious signaling source candidate to give rise to positional information for the striping pattern. But the problem with these chimeras is that they were using chicks that do not display any striped pattern. So we kind of reinvented an old wheel, if you wish, and instead of making quail chick chimeras, we made quail pheasant chimeras, because the pheasants, they have these stripes. So this appears a bit trivial, just said like that, but it was actually a pretty big technical challenge, and I don't think it would have been possible without Nico pairing up with Camille Curance, who is a grad student in the lab and who's an absolute master in micromanipulation. So together, Nico and Camille, they have replaced the half of the neural tube of a quail, of a pheasant by that of a quail. So you can see here these chimeras. After 3 days, you can see that half of the neural tube is stained by a quail-specific marker. And if we zoom in here in this area, you can see that the quail cells have invaded the epidermis and the dermis. They behave like pigment cells, and actually we showed that they were pigment cells. So now we tested, obviously, the expression of agouti after grafting. And what we saw in this graft was pretty much nothing. So I should say that the level of the graft is always made just right here, below the wing level. So there was no expression of agouti, much like the host, but unlike the donor, because the quails at this stage, they already express agouti in bands. So the neural tube does not seem to control agouti expression. What about color? Well, we made long-term chimeras, so 11 days after. We could see that in the grafted area, the color pattern was that of the host and different from that of the donor. So the neural tube does not control the formation of stripes. So we moved to another candidate tissue, which was the somite. Why the somite? Because if you compare here all these different species, on sections you have the early expression of the lateral band of agouti, and you can see that in all the species, it is expressed in the dermis located right above the dorsal lip of the differentiating somite. And the somite was also a very good candidate because it gives rise to the dermal cell lineage, in addition to bones and muscles. And it was shown to be responsible for the patterning of the feather follicles. So this time, Nico and Camille grafted somites of quail in place of those of the pheasant. You can see here the graft 3 days after, so you can see that the quail cells have invaded the mesoderm in the host. And then they tested for the expression of agouti, and ta-da, here it is. You can see that they were able to get a very nice expression of agouti, much like in the donor, but unlike the host. It's pretty clear here on section that we have a quail-like expression of agouti in the grafted side, but not in the ungrafted side. So signals from the somite control agouti expression. What about color as a consequence? So here in the chimera 11 days after, you can see the extent of the graft. And in the area that was grafted, we could see that there was a different color on the patch of feathers. So we analyzed this, like I showed you before, by looking at these lines of feather rows along the dorsomedial axis. So just as a reminder, this is the case of the quail, the color code, black, black, 3 yellows, 2 black, 1 yellow. This is for the pheasant, so 3 black, 7 yellow. This is for the grafted side of the chimera. So you can see that the color code was 2 black, 3 yellow, 2 black, 1 yellow, so exactly the same as that of the Japanese quail. Whereas on the ungrafted side, we reproduced the we still had the same color pattern as in the host. So on the grafted side, we recapitulated with this grafting the color pattern seen in the donor, showing that the somite instructs the formation of the striped pattern. Okay, so if I summarize the results that I have showed to you today, I think we have results saying that the process of periodic stripe patterning is a two-step mechanism. First step, there is an early landmark, which is the mesoderm, the somite, that sets the position of agouti, and thus the position of stripes. And this is consistent with the fact that we don't observe a lot of variation in stripe position in species. So it's consistent with the fact that this early landmark is a constraint to evolution. And then second step, at its given location, agouti controls the duration of the pulse and thereby refines the width of each stripe. And this is what evolution seems to be acting upon. So where are we going now from here? Of course, we want to find out which molecules are involved. And for the first time in this talk, I'm going to talk about morphogens. Sorry, it was a bit late. But we want to know, obviously, what acts in the somite to control the expression of agouti. And there is a bunch of morphogens that are really good candidates, such as Wnt11, expressed in the medial lip of the somite. We've seen differential expression of Wnt11 in some galliform species. So we'll continue along that line, making that screen, that comparative screen for different genes expressed in the somites. Second, of course, we want to continue working on the origin of signals, because today I explained this, right? The formation of the central and lateral yellow stripes. But in many species, there's additional lateral stripes. And in these species, we never see additional lateral expression of agouti in more stripes. So there's something else going on. And this is the project of a master student who joined the lab, Pierre Galipeau. And instead of focusing on galliform birds, he's taken it to another level by looking at ratids, which is the family of ostriches. And here what you have is a baby emu. And you can see that they have this very, very nice and complex striped pattern. And so Pierre has been describing this phenotype and actually doing a bunch of preliminary embryological work to show, at least his first results seem to suggest that the pattern is a combination of early developmental landmark followed by self-organization of the skin. And then finally, we hope to use our data to adjust the way we do modeling of the periodic patterning in the skin. The idea is that within a given frame of simulation, instead of just feeding the computer with stochastic equations, such as Turing's reaction diffusion or chemotaxis and so on, we will constrain this using initial conditions that correspond to developmental landmarks. And so Richard and Pierre have done that in the emu, and they are able to very nicely reproduce its pattern, and they've showed that these initial conditions are extremely important for the timing of differentiation of the tract and the color, as well as for the final robustness of this pattern. Okay, I'm done here. I just need to thank the people of my lab. So this is our lab selfie. I just took that 5 minutes ago. I mean, just 5 minutes before this talk. So I told you about the work of Nicolas Hoppé and Camille Curance. I really want to thank you warmly, guys, because the dream of an Evo-Devo biologist is to transform experimentally one species into another. And they've done that through a huge amount of work. So really, thank you. I didn't have the time to mention the work of Magdalena Hidalgo, who is another postdoc in the lab. She's working on the patterning of color in another family of birds, the Estrelid finches. Richard is also working on the morphogenesis of the skin and is generating computer simulations to reproduce feather tract dynamics. And Camille is also looking at these aspects, but by studying variation, using imaging in different groups, such as galliforms, but also ratids and penguins even. Pierre is working on ratids, as I was saying. We have invaluable technical help from Nicole Quenegdu and Carole Desmarquet, who is a welcome new addition to the lab, and Héloïse Bouvet is taking care of our animals. And with this, I'll thank you for your attention.